Before I get into the details of the Calvin cycle, I want to close the discussion of the photosynthesis electron transport chain with some discussion of the origins of photosynthesis. It is well accepted that photosynthesizing eukaryotes, plants, algae, and some protozoans, acquired photosynthesis by endosymbiosis with cyanobacteria. The figure shows the three-domain taxonomy of life: bacteria, archaea, and eukaryote, with photosynthesizing taxa highlighted in pink and green. The taxa highlighted in green only have photosystem 1. The taxa highlighted in pink only have photosystem 2. The taxa highlighted in green and pink have both photosystems 1 and 2. The path by which eukaryotes acquired photosynthesis by endosymbiosis with cyanobacteria is traced by the red arrow. Note, the archaea have no photosynthesizing taxa. To repeat, the figure shows a taxonomy tree suggesting eukaryote chloroplasts originated in the bacteria branch of life. The green indicates the group has photosystem 1. The pink indicates the group has photosystem 2. Archaea have no photosynthesizing species. Among the photoautotroph bacteria, green sulfur bacteria Chlorobiaceae lack photosystem 2, but make use of photosystem 1 in their photosynthesis process. Purple proteobacteria Rhodocyclaceae have photosystem 2, but they lack photosystem 1. Chloroflexibacteria also have photosystem 2, but they lack photosystem 1. Cyanobacteria use both photosystems 1 and 2. There is no proof positive, but because they lack one of the photosystems, it is believed that green sulfur bacteria and purple bacteria existed before cyanobacteria. Cyanobacteria may have developed from green or purple bacteria, and then acquired both photosystems through DNA transfer or by endosymbiosis with a pre-existing bacteria species to get both photosystems. All three bacteria groups use the VATP ACE rotor to regulate their internal pH. The ATP synthase motor of some type is ancient and exists in all life on Earth. The figure shows the photosystem of green sulfur bacteria resembles the photosystem 1 of plants. Light energy is used to split hydrogen sulfide H2S instead of water H2O. Ferredoxin FD is used to carry electrons to NADP reductase to transform NADP+ into NADPH. Two pathways for the development of the photosystems in bacteria have been proposed. Both photosystems contain chlorophyll and carotenoids in their photon harvesting antenna. Both photosystems include the same or similar proteins, enzymes, and cofactors. The proposal is that photosystems 1 and 2 evolved from a common ancestor that had a proto-photosystem called photosystem 1.5. One group of bacteria evolved the photosystem 1.5 into photosystem 1. Green sulfur bacteria may have taken this path. A second group of bacteria evolved photosystem 1.5 into photosystem 2. Purple proteobacteria may have taken this path. At some point, a proto-cyanobacteria, which have both photosystems 1 and 2, may have merged with or incorporated both photosystems 1 and 2 from ancestral purple and green bacteria by gene transfer or endosymbiosis. The alternative pathway would be the common ancestor of cyanobacteria, green sulfur bacteria, and purple bacteria had both photosystems 1 and 2. At some point, the ancestors of green sulfur bacteria retained photosystem 1 but lost photosystem 2. The ancestors of purple bacteria retained photosystem 2 but lost photosystem 1. The ancestors of cyanobacteria retained both photosystems 1 and 2. Arguments for both of these pathways have their supporters and detractors. Unfortunately, conclusive evidence for support of either evolutionary scenario is lacking in the chemical and fossil record. Neither of these scenarios can make any strides to showing how chlorophyll came to be used by life as the key molecule around which all photosystems appear to be based. In my own thinking, I can see the possibility of green bacteria developing an endosymbiotic relationship with purple bacteria, with the result being proto-cyanobacteria with both photosystems. On the other hand, I do not discount loss of functions. For example, snakes and dolphins have lost limbs. In humans, our tail, appendix, and tonsils all have lost function that some ancestor may have needed. Many parasites have lost all functions that would make them independent of their host. So, in their own respective environmental niches, green bacteria have no need for photosystem 1, and purple bacteria have no need for photosystem 2. If their ancestors ever had both systems, the then unused photosystem could disappear without a trace in a few million years without any detriment to living green sulfur or purple bacteria. I can make no speculations on how chlorophyll came to be discovered by bacteria. Chlorophyll does not occur naturally. Chlorophyll is manufactured by bacteria or by plant chloroplast organelles that were once bacteria. The recipe for chlorophyll is coded in bacterial DNA. Many microbiologists have tried to uncover the most ancient genetic sequence for chlorophyll in the DNA of bacteria, including Peter Lockhart and Anthony Larkham, both at Massey University in New Zealand. In the results of the Lockhart-Larkham DNA study, the authors proposed there existed an ancestral bacterium that used protochlorophyllide pigment for photosynthesis 3.5 to 4 billion years ago. Specifics of how this proposed ancestral photosynthetic bacterium acquired its ability to make this protochlorophyllide pigment are obscure and may always remain unknown to science. In modern plants, chloroplast DNAs are typically 120,000 to 170,000 base pairs long. Chloroplast organelles in plant species have anywhere from 60 to 100 genes in their DNA sequence. This gene count is down sharply from wild cyanobacteria that may have over 1,500 genes in their DNA sequence. The sharp decline in gene count in chloroplast DNA is a strong indicator of their dependence on the host plant for many of the chloroplast's functions. The 1,500 genes in the wild cyanobacteria DNA allows them to make every chemical they need to survive independently. I have not found any reference to how many of the 60 to 100 genes in the chloroplast DNA are responsible for its ability to make chlorophyll. I do see in the figure where geneticists have identified gene sequences that code for photosystems 1 and 2. As both photosystems use chlorophyll, I can assume that chlorophyll is coded for in these photosystem gene sequences. Or at least the proteins that manufacture chlorophyll are coded for. If chlorophyll is coded for in these gene sequences, the question becomes, how did the code for chlorophyll get into the DNA of photosynthesizing organisms? This is a very important question, as 99.9% of all life on Earth depends directly or indirectly on chlorophyll's ability to convert sunlight into bioenergy, ATP and glucose. Also, in the figure, there are gene sequences labeled for proteins and enzymes that are required in the electron transport chain, such as cytochromes and ATP synthase, and others that are required in the Calvin cycle, such as NADH dehydrogenase and rubisco. Remember rubisco, as this enzyme will be a topic of further discussion. Chloroplasts are often compared to mitochondria. Both are organelles of eukaryote cells that may have once been independently living bacteria species. Both have similar, but not the same, electron transport chains. The table compares the chemical similarities between electron transport chains of chloroplasts and mitochondria. Chemical similarities suggest, but do not prove, the electron transport chains in both organelles have a common ancestor. Both chloroplasts and mitochondria were originally thought to be descended from independent living bacteria. The wild ancestors of mitochondria, the alpha proteobacteria, must have existed at least 2.3 billion years ago. These alpha proteobacteria are thought to have been endosymbiotically subsumed into proto-eukaryotes between 2.3 and 1.8 billion years ago. In the time since, the endosymbiotic alpha proteobacteria have lost most of their non-electron transport genes to become the totally dependent mitochondria organelles we see in modern eukaryotes. All animals and plants have mitochondria, but only plants, algae, and a few protozoans have chloroplasts. Chloroplasts are thought to be descended from wild living cyanobacteria. Cyanobacteria can date their lineage back to at least 3 billion and maybe 3.5 billion years. Alpha proteobacteria do not use photosynthesis. So, any common ancestor to mitochondria and chloroplasts must have lived more than 3.5 billion years ago, before the cyanobacteria ancestor acquired chlorophyll and photosynthesis. These cyanobacteria ancestors of chloroplasts may have developed their endosymbiotic relationships with eukaryotes between 1.8 billion and 600 million years ago. The eukaryotes that acquired photosynthesis through their cyanobacteria endosymbionts became plants and algae. The cyanobacteria endosymbionts lost much of their own DNA to become the dependent chloroplast organelles we see in modern plants, algae, and a few protozoan species. The ancestors of alpha proteobacteria had the electron transport chain before oxygen became common in the atmosphere 2.5 billion years ago. Maybe 3.5 billion years ago, the ancestors of alpha proteobacteria were using nitrate NO3 or nitrite NO2 as the final electron acceptor for protein complex 4 of their electron transport chain instead of oxygen. We can search mitochondrial DNA and chloroplast DNA for where the electron transport chains are coded. In humans, mitochondria have 16,000 base pairs in their DNA and only 37 genes that code for 2 ribosomal RNA, 22 transfer RNAs, and 13 proteins. The wild alpha proteobacteria, rickettsia, that could be the ancestors of mitochondria, have 1.1 million base pairs that code for over 800 genes and can make as many as 800 proteins. In plants, chloroplasts have 120,000 to 170,000 base pairs and 60 to 100 genes that code for 60 to 100 proteins. The wild cyanobacteria that are believed to be ancestors to chloroplasts have 3.5 million base pairs that code for 3,000 genes and potentially make 3,000 proteins. Both chloroplasts and mitochondria have circular-shaped DNA strands that contain their entire genome. Circular-shaped DNA are typical of most bacteria. The chloroplast DNA still retains many more genes than the mitochondria DNA. 80 genes for the chloroplast versus 37 genes for the mitochondria. The figure shows the DNA of the mitochondria. The mitochondria genome is color-coded by which sections of the DNA code correspond to proteins that build the structures and enzymes for 3 of the 4 complexes, 1, 3, 4, of the mitochondrial electron transport chain. The genes associated with complex 5 or ATP synthase are also labeled on the mitochondrial DNA. Many of the other proteins that the mitochondria needs are manufactured in the eukaryote cell, from instructions stored on the eukaryote DNA strand. The nuclear DNA of eukaryote cells have billions of base pairs. The human DNA molecule has 3 billion base pairs and a gene count in the range of between 50,000 and 100,000 genes. The figure shows the DNA of a plant chloroplast, in this case, the tobacco plant. The DNA of the chloroplast retains 60 to 100 genes. This contrasts with the 1,500 to 3,000 genes available to wild cyanobacteria. The DNA of the chloroplast cannot be broken apart as neatly as the mitochondrial DNA. Still, we can see in the figure that there are sections of the chloroplast genome that can be labeled for its photosynthetic electron chain. There is a gene section coding for photosystem 1 and a separate section for photosystem 2. There is a DNA section for the cytochrome. There is a DNA section for coding the ATP synthase rotor. There are also DNA sections for coding different enzymes including NADH dehydrogenase, acetyl-CoA carboxylase, and rubisco. Rubisco is an important enzyme in the Calvin cycle. There are other proteins that a chloroplast needs that are coded in the nuclear DNA of the eukaryote plant cell. And that concludes my take on the origins of photosynthesis. In my next video, I will focus on the Calvin cycle of photosynthesis, where the hydrogen split from the H2O by the chlorophyll is joined to CO2 to make glucose.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.